Hello, I'm Aref Shahini, and I'm going to present our work that was published in Science Advances last year. A lot more results than what I'm going to present right now can be found in the website of Science Advances. And let me share my PowerPoint slides. Hopefully you can see it now. So as you know, skeletal muscle loss and frailty is a major medical problem in the aging population. And as we age, we lose our skeletal muscle mass that's important for carrying everyday tasks and also regulating our metabolism. Skeletal muscle is composed of multinucleated myofibers, and there are stem cells that are located within the basal lamina of these skeletal muscle myofibers that are responsible for regenerating them upon an injury. So they are capable of proliferating, migrating, and differentiating into multinucleated myofibers. However, that capacity is decreased by aging. So we wanted to know how can we potentially recover that capacity. If we compare a young muscle to an old muscle, we observe that the total number of myogenic progenitors decreases as we age. Here, staining has been performed for laminin as well as PAX7 to look at the periphery of the myofibers, and the staining for PAX7 shows those myogenic progenitors in the skeletal muscle, and the number of them decreases in the old skeletal muscle. Also, a lot of them become positive for senescence-associated beta-galactosidase, which is a very well-known mark for cellular senescence. When an sperm and egg fuse together to form a zygote, the age is being set to 0. And also, when the cells are being reprogrammed to pluripotency, the induced pluripotent stem cells are having an age of 0. And really, they are staying in that state as long as they are pluripotent. The master regulator of pluripotency is called NANOG. And so we hypothesize that maybe overexpression of NANOG in the cells can indeed give them a useful phenotype without pushing them to full pluripotency. So by overexpression of NANOG, we could induce a partial reprogramming to reverse aging, but don't lose the cellular phenotype, that fate of the cell. So we used a mouse model of progeria called lamin A knocked in. More details about it can be found in the paper of its developer. And expression of progerin in these animals results into nuclear membrane abnormalities and nuclear blips that results ultimately in genomic instability and DNA damage. Let me put my laser pointer. As you can see, the comparison of a wild type to a lackey at 1 year of age, the lackey has been quite old, and this phenotype is what we use to investigate the effect of NANOG on aging. These are heterozygous lackey animals. The homozygous lackey animals, they actually die at the age of 3 to 4 months. We use the heterozygous animals for our experiments. We crossed lackey animals with NANOG expressing animals. So the final genotype is capable of NANOG expression by administration of doxycycline, and it also has the progerin that induces that pro-aging phenotype. So in this fast aging model, we express NANOG by injection of doxycycline into the TA muscle. We observe that expression of NANOG is observed in all of the cells, including those myogenic progenitors stained for PAX7, and that's red in here. We could measure the number of those PAX7 myogenic progenitors by performing immunostaining for laminin, as well as PAX7 positivity. So we observe that the number of those myogenic progenitors are decreased in the case of lackey, and then increased back up by NANOG expression. Each bubble here shows an animal, and we have cut the muscle at multiple different sections and performed immunostaining to make sure we have a good representation of these calculations. So a lot of statistics goes into this. And also expression levels of senescence associated beta-galactosidase decreases in the skeletal muscle by NANOG expression. So these two observations suggest that in vivo or in an animal, expression of NANOG in the skeletal muscle decreases that senescent phenotype or reverses aging in those muscle progenitors. We also looked at formation of new myofibers by performing immunostaining for desmin as well as embryonic myosin heavy chain that are marking those newly formed myofibers or the fibers that are in the process of regeneration. And so we observed that expression of NANOG significantly increases the number of those myofibers, suggesting that maybe due to the increase in the number of those myogenic progenitors that I showed you in the previous slide, they also form new myofibers in the case of NANOG expression as well. We looked at the hallmarks of aging in these cells upon NANOG expression. To do that, we first isolated the myogenic progenitors from 25-year-old female, 75-year-old female, and 68-year-old male, so 3 independent biological repeats. And we overexpressed NANOG by transducing the cells with lentiviruses carrying NANOG. And NANOG could be overexpressed in the cells by an addition of doxycycline in the media. So DOX stands for doxycycline. By addition of doxycycline in the media, we observe a expression of NANOG that's similar to embryonic stem cells, and that increases the proliferation capacity of senescent cells. The case of senescence here is replicative senescence, meaning that we have passed the cells in culture for multiple population doublings for about 2 months. So the cells are becoming senescent and they're becoming positive for SA beta gal. And here we show that NANOG indeed is increasing the proliferation level, but it doesn't give them immortality. So we also looked at the RNA sequencing to assess the transcriptome of the cells, and the gene ontology clearly showed us that the levels of the DNA repair and replication, as well as proteostasis and mitochondrial function, are recovered by NANOG expression for 5 days, 10 days, or 15 days. And so here we are comparing them to the senescent phenotype and the young versus senescent comparisons and pathways. And the normalized enrichment score, if it is positive, that means that pathway is being enriched by NANOG expression. So we further wanted to understand what if these changes in the expression levels are actually functional, and if we see a function at the levels as well. So the first hallmark that we assessed was genomic instability and the DNA repair. So genomic stability is very much related to the levels of heterochromatins in the cells. Heterochromatins are those compact sites of the nucleus or genome that are responsible for preventing transcriptional noise. The transcriptional noise increases as the cells age due to the loss of heterochromatin. Here we performed immunostaining for H3K27-methyl-3, which is a mark for heterochromatins, and clearly that's being decreased in the cellular with cellular senescence, and expression of NANOG increases that back up to the young status. So also cytoplasmic chromatin fragments shown by arrows here are also being formed in the senescent phenotype due to the dislodge of those parts of the DNA that are heterochromatin rich, and they are being dislodged from the nucleus due to genomic instability, and they go for autophagy. Expression of NANOG is essential to these effects because after removal of NANOG for 14 days, those effects go away. So NANOG does not induce immortality in these cells, and the presence of it is essential to the effects that we see. We also performed immunostaining for gamma H2AX, a very well-known mark for DNA damage, that is the phosphorylation of H2A histone due to ATM activation. And what we observe here is that the senescent cells are having a higher level of gamma H2AX, which could be due to the higher level of DNA double-strand break in the cells. And expression of NANOG for 15 days decreases that back down to the young level. Quantification shows here. And each bubble here represents a donor. So each bubble is independent repeat using a different donor. The transcriptomic analysis showed restoration of DNA repair pathways, and we wanted to further investigate this using functional assays. So we performed COMET assay, or single cell gel electrophoresis, in which we treated the cells with a very basic buffer. And then we performed electrophoresis in that basic buffer. So the DNA double-strand breaks are being pulled away from the genome, and that's being shown as a tail by staining for DNA dye. And we can measure tail DNA % as a metric for double-strand breaks. The levels of it is increased in senescence and then decreased back down by NANOG expression. We also performed a challenge experiment in which we treated the cells with etoposide. Etoposide inhibits the ligating arm of topoisomerase type 2. Topoisomerase type 2 is responsible for releasing coiled coils in the DNA, and it performs cuts, and then the coiled coil releases itself, and then it ligates back. But when we inhibit the ligating arm, then there are cuts being made into the DNA, and that results into the activation of the DNA repair pathways, such as ATM. When we look at the levels of ATM and the phosphorylated levels of ATM prior or after etoposide treatment using Western blot, we observe that the total levels of ATM is increased by NANOG expression, and that results into the activation of the ATM upon etoposide treatment. Further down the road, that can phosphorylate H2A to form H2AX. And as you can see, after the etoposide treatment, the H2AX is much higher in the case of young and NANOG expressing cells. So ATM pathway could be activated in these cells. And when we also performed this analysis, we looked at the single cell immunostaining for H2AX, gamma H2AX, as well as tail DNA %. We observed that prior to etoposide treatment, the tail DNA % is lower in NANOG expressing cells. However, when we treat them with etoposide for 2 hours, we observe that the cells are having the similar levels of double-strand breaks, but a lot higher levels of gamma H2AX. So in response to the same levels of damage, the cells are having a higher levels of gamma H2AX, suggesting a higher activation of DNA damage response that can be measured by the fold increase in the gamma H2AX intensity upon 2 hours of etoposide treatment. And here the hours are shown as hours after etoposide. So during this time, the cells are being treated with etoposide for 2 hours, and then etoposide is removed from the media for 2 hours and 12 hours. And you can see that in the case of young and NANOG expressing cells, they quickly recover from that DNA damage, and they fix their double-strand breaks. However, the senescent cells are incapable of doing that with the same pace. We also looked at autophagy, and proteostasis, or transcriptional analysis, showed clearly that transcription, translation, and ribosomes are recovered by NANOG expression, as well as protein folding. And we wanted to also look at the autophagy as well to see if that's being changed. The first observation that we had was that the levels of senescence-associated beta-galactosidase is decreased by NANOG expression. That's a beta-galactosidase which resides into lysosomes. And sure enough, when we looked at the lysosomes using lysotracker that binds to the acidic organelles inside the cell, we observed that expression of NANOG decreases the amount of the lysosomes in the cell. And that results into decrease in the protein content and cellular radius as well. To really look at the autophagy flux and measure that, we treated the cells with chloroquine and starved them. So when we starve the cells are trying to break down some of the proteins to produce some energy for themselves. And that's going to go through autophagy. However, chloroquine is going to be inhibiting that process of fusion of autophagosomes to lysosomes. Therefore, there is accumulation of autophagosomes that can be measured using flow cytometry for cyto-ID dye. And that shift in that intensity can be measured as a metric for autophagy flux. So as you can see here, the autophagy flux is increased by NANOG expression. And that can be due to the activation of mTOR or AMP kinase. And as you can see in this Western blot, neither phosphorylated levels of mTOR at serine 2448 or the total levels of mTOR are changed by NANOG. So none of those are changed, but the levels of AMP kinase as well as the phosphorylated levels of AMP kinase, they are being recovered by NANOG expression in the senescent cells. And the quantification clearly shows a significant increase by NANOG expression in the AMP kinase, and the senescent cells are losing those levels of AMP kinase. So we hypothesized that AMP kinase is the essential factor in this case, and therefore we inhibited AMP kinase by addition of chemicals that inhibit this kinase. We observed that autophagy flux is decreased back down to the senescent levels by addition of these inhibitors. The levels of phosphorylated levels of AMP kinase goes down, and that results into increase in the cellular radius and also decrease in the mitotracker. Mitotracker is a dye that binds to the mitochondria with membrane potential. So if the mitochondria have higher membrane potential, there is a higher intensity of mitotracker, and that's being observed by NANOG expression. And that's clearly dependent upon AMP kinase activity, because in the case of AMP kinase inhibition, that goes away. ATP concentration is also increased by NANOG expression, and that's decreased back down by CC and SDI. So all of the ATP concentrations are being reported here as nanomole per milligram protein. So they are normalized to the cellular size. When we measure the total levels of the ATP, the total levels of ATP actually increases in senescent cells as compared to the young cells. And so here we are always normalizing ATP to the protein content. And another staining here being shown for mitotracker, you can see that from the young to senescence, the intensity of mitotracker decreases and NANOG increases that back up, suggesting a higher mitochondrial membrane potential in NANOG and young cells. And interestingly enough, the levels of the mitochondrial DNA over nuclear DNA is decreased by NANOG expression, and which was increased by senescence. So really, the senescent cells have a lot of dysfunctional mitochondria that have a small membrane potential, and these mitochondria are not capable of generating ATP for the cell. So we thought that maybe the mitochondrial quality control proteins, PINK1 and Parkin, might be playing a role in this case. And we performed Western blotting for Parkin as well as PINK1. We observed that the total levels of Parkin is recovered by NANOG expression, and the truncated form of PINK1 is also restored in the case of SN15 as well. That suggests that the process of PINK1 that is being imported into the mitochondria with membrane potential is being recovered. Then PINK1, after being imported into the mitochondria with membrane potential, is being cleaved and it's sent for degradation. However, in a dysfunctional mitochondria, PINK1 accumulates on the membrane, phosphorylates Parkin, and that results into ubiquitination of the mitochondria with lower membrane potential. And that sends the dysfunctional mitochondria to mitophagy. So with this system, by overexpression of NANOG, we observe a higher levels of Parkin. Therefore, those dysfunctional mitochondria are being sent to mitophagy. And as I showed in the previous slide, the autophagy flux is upregulated. So the cell is really clearing up those dysfunctional mitochondria to form new mitochondria. and induced mitochondrial biogenesis essentially create new space for new mitochondria and increase the levels of ATP in the cell ultimately. We look at mitochondrial respiration and electron transport chain using a test called mitostress test. We treated the cells with oligomycin, and that results into inhibition of complex 5. Therefore, all of the hydrogen protons are accumulated in the inner membrane space. Then, by treating the cells with FCCP, all of the hydrogen protons are being moved downstream, and that results into maximum electron flux and maximum oxygen consumption rate. Antomycin and rotenone shuts down the mitochondria by inhibiting complex 1 and 3. So we can measure basal, reserved, ATP-linked, and maximal levels of respiration with this technique, and all of those parameters are being restored by NANOG expression. Overall, in this work, we showed that expression of NANOG in senescent cells results into a rejuvenated phenotype. The cells gain back their cellular size and morphology, and also they have an improvement in their mitochondrial health and cellular energetics. The nuclear integrity and DNA damage response is restored by NANOG expression, and that results into improvements in genomic stability and cellular proliferation in the senescent cells, both in vitro and in vivo. That results into formation of de novo myofibers in the skeletal muscle. With that, I would like to thank our collaborators and my PhD advisor, and also the funding agencies. And thank you for watching.